So our final speaker in this session is Dr. Julian Hiscox. Dr. Hiscox been working on coronaviruses for the past 3 years, including on an FDA-funded international program looking at medical countermeasures or information to support the development of medical countermeasures. And he will be speaking about AI virus prediction.
All right, good afternoon all. Thank you to the organisers, one of which was me, for the opportunity for speaking today. And it's a pleasure to follow Reby and Jim and co. I think one of the things, as we were alluding to earlier after the presentation on Hiscox, is I guess the pandemic response was a system of speed dating, in that many of us worked with people that we'd never actually worked with before and did it very well. So this is the first time I've ever met Jim in person, and the speed date was via Miles at the back. We'd been working together for 3 years now. So I'm very much a biologist. I'm very much a Steve Jobs type group leader, where I will place impossible demands upon my group and say, we've got this really good idea, just make it happen. And somehow magically the magic occurs, right? So I'd like to tell you about our story on using machine learning to perhaps predict the future of viral evolution. And this is something we've been discussing with for the past 3, 4, 5 years now with our funders and colleagues at FDA, Tracy being one of those. And perhaps we're gradually getting to a realization of this now. So I had a FDA-funded consortia that was funded right at the beginning of the outbreak through emergency funding from FDA to Miles as part of our Ebola work. I should say that I have been working on coronaviruses for over 30 years as well, so I'm not an interloper like Arnab to the coronavirus field. And we kind of... Yeah, I know, Callum, yeah. So we split our FDA contracts into several different areas, and the one I was particularly interested in was using transcriptomic approaches, underpinned perhaps, as I would say in this talk, by machine learning, to look at functional viral or functional host biology. And we're very much able to do all of this through all of the biomaterials that we received on the right-hand side through various funded projects and organizations as well. So I'm going to tell you a little bit about viral population genetics. I too did a degree in genetics like Jim, apart from I did it at a far superior institution of University College London, in a far grimmer place of London and with not a nice view as Aberystwyth. But when we consider a virus, right, we've heard a lot about viral genome sequencing in the outbreak. Many of us have done it as part of our own laboratories or big national entities. And what you tend to return and what you tend to see is a consensus level sequence. But underneath that, like Jim was saying with proteins, is an awful lot of viral genetic noise. And that's something that Miles and I have been working on for many years now with Ebola virus, is working out how this genetic noise turns into an evolutionary force, or how evolutionary forces act on this genetic noise to do something to the virus under some type of selection pressure. We're particularly interested in medical countermeasures, but this could be driven by non-pharmaceutical interventions as well. So your normal sequencing returns your consensus sequence, but underneath that then is a whole array of variability. And if we think of our classical genetics, those changes can be, and mostly will be, deleterious, or they will be neutral, or in some cases they will be advantageous under a selection pressure that allows that virus basically to grow better than its competitors. That's all Darwinian evolution is about. Can I make it to the top of the pile better than everybody else? And if I can do that, then I expand within a particular population parameter. So we've really done this a lot with Ebola virus and medical countermeasures, and Reby and co in the lab have been doing this with SARS-CoV-2. And as an example of that, one of the first major changes in the SARS-CoV-2 genome was the D614G substitution in spike, right? So everybody got on board the bandwagon for that, quite rightly, and showed that that particular change increased transmissibility of the virus. And me being slightly genetically orientated and probably slightly contrary to, as those of you who know me, to accepted dogma, said, well, isn't this thing in NSP12, the viral RNA-dependent RNA polymerase, the P323L substitution, which everybody, if you read the papers, probably including one of my own reviews actually, was a bystander mutation, right? But what you found was in March, if we look at UK sequencing data, and those site hourglass things give you an overview of the consensus level sequence and the minor variants for D614G and P323L in the UK population at that time and worldwide underneath it. And you see this very, very rapid selection in the population from D614 to G and from P323 to L, and that then defined the lineage B1 virus, if we go via a Pango lineage, which has become the dominant lineage and its descendants worldwide subsequently. It's surpassed everything. And likewise then in the lab, we've been studying viral evolution under selection pressure in antivirals. So if we then think about that NSP12 substitution, not only can you see it occurring rapidly at a population level, you can then go into individuals and look at it. And we were very lucky through ISARIC to get longitudinal samples of patients from early in the outbreak. And again, within a matter of days within an individual, you can see a rapid change, not only from D614G, but also from P323L. So these are probably linked in a genome. And of course, many studies have shown why D614G is important. And Mohanad in our lab has been looking at P323L using proteomics. And we think it's a host adapting change that allows better interaction with host proteins. That's one of our hypotheses for that. And indeed, it was really brought home to us through a more fortuitous route with work that was led by Miles and colleagues at what was Public Health England in the audience, where they were working up animal models for COVID-19 at the beginning of the pandemic by infecting rhesus and cynomolgus macaques. Miles kindly gave us the RNA from those samples and said, sequence them and tell me what's happening. And what we were able to do was to see that the output virus from those animals was different from the input virus. So the input virus that was used was the Victoria isolate of the virus, probably dare one use the phrase Wuhan original, where when you sequence that viral stock, the predominant alleles, NSP12 and spike, were P in NSP12, P323L, P, yeah, in NSP12, and D in position 614 at spike. But what we noticed then in the monkeys when we sequenced those samples given to us by Miles is that there was a very, very rapid swap within days from P323 to 323L. Nothing happened with D614G, no D614G change, but that in monkey selection drove whatever it was about that mutation, drove selection in those monkeys. And in actual fact, we think it's to do with host adaptation. But what it also shows you actually is that maybe that linkage with spike is to do with transmission. But when you have those two substitutions together, exactly like Arnab was showing with N, virologically, I tend to think of virus changes in logs. I'm sure, Paul, you do the same. If it's not a log, then it doesn't count because we plot viral growth in log units of log 10, right? But what these mutations are additive. They might not make a log difference, but they do make a difference to viral growth. So Hannah and I came up with this idea of transmission of these variants. Let's say you're infected with Victoria. where P323 is a very dominant level, somehow that L has to come through. So what we actually think, based on that monkey data and based on sequencing the ISRIC, some of the ISRIC sample set, is that much in classical genetics, you have your founder mutation, but as Callum has eloquently said many, many times, one of the things we need to realize about SARS-CoV-2 is that its window of transmission is very, very short. It's even before symptoms. So you've got 3 or 4 days to spread the virus, and then that's you done. Then you get your symptoms, and then you're told to isolate, not to spread the disease. And I'm sure Callum can wax lyrical on this later and tomorrow. Callum, I'll finish talking, and then then you can rant away. But the point is that your window of transmission is short. So if you get a genetic change in the virus, in normal, quotes, healthy people, that change has to occur fairly quickly and be spread fairly quickly and have some type of advantage at an individual and population in order to be transmitted. So that's kind of what our model reflects. And also sequencing some very, very healthy people, samples given to us by the Ministry of Defence in the UK. Again, in those healthy individuals, we can see quite a large number of changes occurring very early in infection. So you don't really need the immune compromise to get the changes, which is, I think, current accepted dogma for the emergence of VOX. Probably what those immunocompromised people provide is a larger window of infectivity compared to the normal population. And much like Jim was explaining his philosophy behind his work, I'm just explaining our philosophy of viral genetics before I get on to talk about the machine learning. So the other thing we were quite curious about, Xiaofeng, one of the postdocs in my lab, and myself, and many of us discussed this at the beginning of the outbreak, is the sequence of the virus at the Huanan market and what it can tell us about the origins of the virus. I won't get into the origins of the virus, but I certainly am a moderately firm believer that the origin of the COVID-19 pandemic is the market. That's the amplifying event. But when we did it for a laugh, really, to start with, and that's literally what it was, what if, Xiaofeng took the publicly available sequence data from the market vendors and workers at the market and analysed the consensus sequences, as loads of people have done, and also the minor variants. And to our surprise, what we found actually in the minor variants, and this is an example of spike, is that in those minor variants, there was the presence of mutations that we associate with future VOX, right? So what you're almost looking at is God's playbook, if I may use that phrase, for the pandemic, right at the beginning of the pandemic. Now, Callum will be more familiar with this, being an expert also on respiratory syncytial virus, where in the old days, we used to call them hypervariable regions. But by having a look at that sequence, we can identify regions of the genome that will tolerate change and regions of the genome that won't. And by doing very, very extensive deep dive sequencing, you can identify that genetic noise from the background sequencing error rates. And we've continued to develop this philosophy with Tess and Aya in the lab, looking at molnupiravir. Now, molnupiravir is a viral mutagen, and we can see that when we sequence samples from patients as part of the agile response and also in cell culture as well. So, there's definitely selection pressures and things that will cause these viruses to be variable. Some of these molnupiravir genomes will survive and become what I would call a remnant genome. So they go on and persist. So how can we now start examining this at a global population level? How can we start looking at the variability of the virus and predicting that variability and doing useful things with it? So thankfully in the UK, and we were, and I say we, at Liverpool in the largest sense were part of this, were part of COG UK, our national sequencing effort, where we sequence, as Arnab said, some 2 to 3 million viral genomes, and all of those were made publicly available pretty much as soon as they were sequenced. So we were able to dip into that data to start looking at viral variability at a very large population scale. So we sampled, and this is Xiaofeng's work, 100 genomes per day for nearly 3 years. So that's over 96,000 UK patients. And he derived their dominant consensus sequence, let's call it the majority level virus in a person, and also their minor genomic variants. We particularly chose a subset of that data because we wanted everything sequenced by Arctic Illumina on a NovaSeq, but just at different sites in the UK. So the sequencing pipeline was the same. Now, for those of you who do a lot of sequencing, and Paul, myself, and James were laughing the other day that during our PhDs, and Miles was probably the same, we actually sequenced manually, where we would sequence about 300 nucleotides every 4 days. And that was if you were doing a good job. And that was with radioactivity and aluminium backs to stop the heat transfer melting your gels, right? So that was on a good day. Now what we're talking about is analyzing gigabytes and millions upon billions of individual nucleotide positions within a viral population, within a person. And sometimes we also sequence the human transcriptome as well and the microbiome. So all of this computationally absolutely staggers me. So one of the things we did when the outbreak started is that we invested in 4 servers because we were forever chalking up the servers at the university and annoying people. So a lot of this is with a 1,000 core cluster that we've got in our own lab. And so when you think of analyzing sequencing data, again, in the old days, we would get an autorad and we would score the nucleotides on the autorad. Many of you will look blankly at me in the audience because you're all under the age of about 45, right? But that's what we did in the old days. Now, you clever youngsters use scripts and all the rest of it in order to derive sequence information. But when you do that, 200 megabytes of Illumina FASTQ data requires 5 hours per sample in order to get a consensus genome and a minor variant readout. So, and not only that, one of the mind-boggling things to me about sequencing now is that it costs more to store your data than it does to actually sequence. So it requires 1.2 gigabytes of space in order to store that amount of Illumina data from one person. So Xiaofeng came up with a new pipeline that he called AO minor. Don't ask me what it stands for. That takes that computational time to analyze and get one genome sequence or a genome sequence and minor variants from 5 hours to about 21 minutes. And it's available on our GitHub site. And what it effectively does is give you a very nice Excel table that I can understand that tells you the top amino acid position at a particular part along the viral genome stroke proteome, let's call it, its coverage, its read depth, and then also the minor variants underneath that. So when we start then looking at the genetic variability within SARS-CoV-2, within the UK population, over a 3-year period, what we do is compare amino acids with each other at the same part of the genome, because one of the things that we notice with Illumina data is that there's Illumina bias, and Illumina is biased towards the 5' and the 3' ends of the cDNAs that it sequences. And it starts gaining more and more errors as it goes into the middle of a sequence read. So the way to get around that then is to compare vertically rather than horizontally. But again, what we notice in these animations now are amino acid positions within each gene, let's call it, of the SARS-CoV-2 genome, and the different VOCs, Delta, Alpha, Omicron are all colored differently. So our homegrown Alpha variant in the UK is red, although I'm really sure it originated in France and came over in a lorry. The green Delta variant from India is in green, and then blue is Omicron. And what you can see is that many of these amino acid positions change in variability. They're kind of offered up for selection, and some of these then operate under selection pressure as we move in the pandemic, become fixed on the right-hand side, and then move downwards. So also what this tells you actually is that spike is one of the most variable proteins, and has been and will be throughout the pandemic. And so when you plot this then starting at the beginning of the pandemic and going all the way through to December 2021, is that you notice that there is an increase in the variation frequency of SARS-CoV-2. So SARS-CoV-2 is not slowing down in its evolution. It's not becoming static. Its genetic variability is increasing, exactly as we would see for other animal coronaviruses, as we were reminded this morning, such as avian infectious bronchitis virus and porcine coronavirus. The evolutionary trajectory of a coronavirus is to diversify, not to funnel down into a population bottleneck. So how can we start looking at this mathematically? How can we predict what future sites might be variable using machine learning approaches? So again, we turn to a classical genetic approach where we use a W value. So we assign this for each amino acid. So we sequence the nucleotides, we assign them into amino acids, and we look at their variability at an individual level. So a W value of 1 means that that amino acid is fixed in that sequence population. There's very, very little change in it. If there is more change in it in that sequence population, then the W value decreases. So if we take a snapshot of W values, say in the UK in March 2021, what you see for spike, the very top blue circled line, linear going left to right or right to left, is that most sites are not under any selection pressure, they are fixed. And then you get sites in green that are under selection pressure, they are becoming, there's lots of variability associated with those sites. And then there's some sites that have changed, let's say from the original virus, and become fixed in the population, like D614G, like P3G3L, and some of the other markers associated with alpha that was emerging at the time just before Delta came along. But again, what you can see in that data from March 2021, before Omicron was even kind of known about, if I've got my years correctly, is that we're seeing VOCs associated with amino acid positions associated with Omicron under selection pressure in data from the UK. So what that tells me is that it's only a matter of chance where a variant emerges. Nothing particularly unusual about South Africa. It might just be to do with when you get a little bit of waning immunity to a particular variant, then a new variant will come along because it has a very, very slight advantage. And so this is what we then see in this like storybook of spike variability over a 3-year period, where again, you're getting these amino acid sites offered up for selection. This is the bottom part. And then you get, for example, the alpha variant on the right-hand side in red becoming fixed in the UK population. What's interesting to me in these types of things are some of these black positions marked in black that are sort of going towards the right-hand side, but don't become fixed. And then, of course, as the alpha variant was overtaken by the delta variant, we can see delta variant moving through. We've got amino acids associated with the Omicron variant kind of sloshing around there. But it really wasn't until we were hit with Omicron from Southern Africa does it then become predominant in the UK. But certainly the genetic noise in the UK suggests that an Omicron type virus could have equally emerged in the UK as it did potentially in Southern Africa. So how can we then use machine learning? This is our machine learning pipeline, and I will skip over it because more clever people in the lab understand this than me. But effectively, what we're looking at is the variability in amino acid position and its correlation between how it becomes fixed in the UK population over a certain period of time. So we cluster that data based upon amino acid and principal component, mainly it being fixed. So this is a map of changes in spike. So I'll take you through this. So this is only spike. Each dot on that, and ignore the red dots at the moment, is an amino acid position in spike of SARS-CoV-2. So if we take 12 months of data at 100 genomes per day, using that machine learning approach, we can identify what sites are most variable in spike. These are illustrated in blue. So we would predict that if changes were to occur, they would be more likely to occur in the blue cluster than the orange cluster than the grey cluster. So imagine you're 1 year into the pandemic and you decide you're going to skip the pandemic, you go into suspended animation, or you travel at the speed of light for whatever period of time, and you emerge back 2 years later. And you say, okay, what amino acids have now changed? Well, the majority of amino acids that now changed were predicted by this particular model, and those are in red. So the model predicts with about 87% certainty what amino acids would have changed in spike. And that's based on the first 12 months of data. We can then take the 36 months worth of data and look at what amino acids are likely to change in the future. And of course, then you can map these amino acids onto bits of the spike glycoprotein because we have its crystal structure. So you can say, for example, some of these changes will occur in the receptor binding domain. So you will expect changes in receptor binding. So basically, the longer you leave these models, the better, and it's obvious really, the better they perform. But we're at a stage now where it's possible to predict what sites are likely to be most variable in the future. So of course, as some of you may know, CEPI have issued this challenge called the 100 days. How can you shut a pandemic down in 100 days or introduce medical countermeasures? So we think this type of work will help towards this because it will tell you what sites within a viral genome may change and which will remain fixed. So if you're thinking of medical countermeasures like vaccines or antivirals, that becomes particularly important, and also diagnostics as well, because one of the problems we had at the beginning of the pandemic is that these WHO primer binding sites that are in the envelope changed as the virus changed. So again, if you can predict the future, it gives you longevity of a response to the pandemic, and it allows you that more rational vaccine design or more rational monoclonal antibody design. So one of the things we would like to look at, and we would look at the moment, is that, of course, we had the luxury of having 3 years of highly sequenced data where we sampled it 100 genomes per day. But if you had 500 genomes for a week, would that be enough? What limits would you need in order to get that data to inform a 100-day mission? And of course, would this work for other viruses such as Ebola virus, which is one of our other major interests in the laboratory? So I'll end there just by thanking all of my current and previous group. You see, Reba, you are now off the list. My great shame. And pretty much this is very much a team effort, but very much grateful to our major funders, the FDA, the Defence Lab, and various other UK funders and European funders as well. And again, we couldn't do this work without all that sharing of samples that occurred throughout the pandemic. I'll end there, take any questions, and conscious we need to get coffee, tea, and toilet breaks in.
Thank you, Julian. So can you, in the absence of time, can you speed this up for a new virus that may occur in an in vitro process?
Yeah, potentially. So I guess the thing is in vitro is that you lack a selection pressure. Your selection pressure is the cell that you use. Goes back to Jim's point that there are hundreds of thousands of proteins and they all vary in different cells, right? It's exactly the same with a virus. So the virus sequence that comes out of an A549 cell might be different to a callo cell, which is where then ex vivo models might come in. But I think one of the things with the 100,000 genomes is that the selection pressure that you see in a population is what you will see.
What about the selection pressure of the immune environment as the pandemic progresses or the outbreak progresses?
Well, so one of the ideas from that, and I will now steal it, is then thinking about what occurs in an animal model, right? Because then you are getting an immunological selection pressure. But, you know, again, I think this question now is 10 genomes a day, 100 genomes per day, 1,000. At what point, when do you reach that limit, that prediction limit?
Thank you.
Probably loud enough without. Can you comment on what vaccines do to the selection pressures for the variants that you see?
Yeah, absolutely. So again, I mean, Callum, I know, used to bang his head about this, but you would get people saying, well, we can't use the vaccine because the virus would change, right? Which is such a nonsense argument, right? You can't even describe it. But so you have to use the vaccine, right? Because that is reducing the severity of disease, as Reeby's slide showed, which I'm now going to steal because it's better than the one I use, right? But the whole point of the vaccine was to reduce disease severity. We've spoken about earlier that a coronavirus vaccine will never stop transmission, right? It just won't. But it reduces disease severity. But of course, then you've got an immunological profile. Let's say you use Wuhan original. You're vaccinated against Wuhan. So when Delta comes along, or Omicron, and we know from our work we've done with collaborators that if you use a Wuhan vaccine, it's no good against Omicron. It just doesn't make a difference. So I think what the vaccines do is select for a slightly different variant. And that's what we're seeing in this data with Spike.
Did you see in those beautiful graphs that you had, which were changing over time, did you see changes in the distribution of the variants according to when the vaccines were introduced?
So that's what we've got to look at. But the complication in the UK is that just about as we were to start our vaccine effort in March 2021, we had 30 to 40,000 people coming back from a religious festival in India spreading Delta, right? That's not their fault. That's our government's fault for not having involuntary quarantine, right? Or just saying no travel, right? So it becomes more complicated to answer that type of question because we've got a very rapid switch. But I think we could look at that type of thing potentially from Alpha to the vaccine, but we were subsumed very quickly with Delta.
And it's a naive sort of hypothetical, but I mean, if you had vaccines against all of the hypervariable regions, I mean, could you sort of squeeze out that possible future change?
I think it would give you a way towards the mythical universal vaccine. And I say mythical because I'm slightly skeptical of it, but certainly it would allow you to identify sites, whether they be T-cell epitopes. I mean, corona vaccines, whatever they are, whatever animal, to do with neutralization, it's all to do with neutralization. It would allow you to identify sites that would be longer lived, if that makes sense.
Yeah, that's great.
Thank you, Julian, and thank you for embarrassing me at various points.
In a nice way.
Yeah, I've got a question for four of you. Some may know that Julian and I are interested in shooting, and one of the things you have to do if you're shooting is follow a target and realize when the target is slowing down to give the chance a shot. One of the problems we've got in this biology is spotting the movement of these data points and identifying the data point becomes stable. For my work, I needed to be able to say with confidence when a risk factor was likely to be important in identifying patients at risk, whether that was obesity or a history of cancer therapy. And when I watched the data, I could see we had essentially chaos. Then you had confidence intervals with a point estimate. And as you watch the data over time, you'd see the confidence intervals getting wobbly and then starting to zoom in. And I felt as soon as the confidence intervals started to stabilize in a particular direction, chances were that the point estimate wasn't going to deviate from without them. But I couldn't find a mathematician or a statistician or bioinformatician that could give me an engineering solution to how to identify when a data point or the confidence around that data point is starting to stabilize. And all your images had exactly the same problem. You could see that we knew it was going to become delta,
Yeah.
but we didn't know it at the time. But that dot was starting to move. So there must be some kind of, when the military probably know how to do it, But there must be some way of saying a data point is starting to track across the screen and then it's starting to slow down. And that surely must be the point we're saying that is now important. So is there something the machine learning people can do?
So from our perspective, Callum, if an amino acid moves into that bottom population, that's when it's offered up for selection. So of course, only some of them make it through to the right-hand side. So these things in black that don't make it through are the potential future ones. But there's so many of them, you really have to do your functional biology now to say what are the implications if we have a change in that area.
I think that's what I'm saying. Is there a challenge that we can make that machine learning people can spot the ones that are moving in a purposeful manner and are then slowing down?
That's what that is. That's what the ones are in blue. But there are lots of them, so you don't know which blue ones are coming next.
Right, so
you just know it's going to be in a blue.
Okay. So you've got that with okay, so that's with hindsight, I suppose. Yeah, but this is predictive.
Yes, it's predictive.
So you are doing it.
Yeah.
Cool.
Julian, my question is in relation when you use the machine learning here, do you also take into account that there is like CPG reduction by cytosine deamination? Because that's another host-directed process that changes the shape of the genomes of RNA viruses.
I'm going to answer you in a very mature manner. La, 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 right? No, we don't, but we have looked at, we've been obsessed with ADAR with Paul for years, or
Apobex.
Yeah, Apobex and nitrous, what's the latest one in the lab? Hannah, was it you? Nitrous oxide, something or other? Reactive oxygen species and so on, also potentially influencing the genome. The answer to your question is no, but we probably should.
Very interesting. I can know fascinating. For this prediction. So like unsupervised and supervised. Clustering is not prediction, but rather proof discovery.
Yes.
Probably tried supervised methods as well.
I'm going to sort of like not look at you. I think this is unsupervised, but you're right. What we're effectively doing is clustering groups of amino acids together that have similar properties. And in this case, the similar property is its variability pattern. So then the things in blue have a particular variability pattern that we would then predict would change.
Yeah. Because I would say that there's a huge space. I mean, not in data gathering, right? But in machine learning that you can exploit, that is probably still, I mean, things that you still can do, right? Using supervised methods, like, I don't know, logistic regression probably being the simplest one, right? But there are many more advanced if you have more data, right? And in that case, I would call that prediction, whereas probably this is more like a group discovery.
Yeah,
right?
yeah. And I think one of the obsessions that we have, to sort of answer your question slightly obliquely, is that one of the things I've always wanted to look at is host transcripts versus what's happening to the virus. And likewise, what Reby and Jim have been showing, you know, you've got a whole load of patient categories. How do they then correlate with what you see in the virus? Hospitalization, shedding, transmission period, all of the rest of it would be good. I think probably it would require another trip to Slovenia to talk to you about that, which I've been more than happy to do. Right, I'll tell you what, Tracy, we were right ending there. Thank you.